Hamed is a PhD in neuroimaging from Tehran University of Medical Sciences and did his postdoc with Martin Paulus for 2 years at the Laureate Institute for Brain Research, Tulsa, Oklahoma. He obtained a position as a research and clinical department of Iranian National Center for Addiction Studies, and he's now an associate investigator at the same institute, working on using fMRI and different brain stimulation with hopes to contribute in reshaping the future of psychiatry and addiction medicine. So, welcome, Dr. Hamed.
Hello, everyone. I'm really happy to be here. I'm really excited and delighted to be here. I met Felipe in his office in 2009.
Yes, at Bet Israel.
But you didn't change at all. I mean, during that time, like 30, I was just thinking, oh my goodness, Felipe is just doing the same.
Stimulation.
You are doing great. So I'm really excited to be here. I'm really happy that we will have time to talk about brain stimulation and potential for brain stimulation. So what I'm going to talk about today is about potentials for individualized transcranial electrical stimulation. As other speakers have mentioned, there are main challenges for that specific issue. So we will try to kind of cover that interesting topic. So let me see, the screen works. Okay, so I don't have any conflict of interest to present. So among different brain stimulation technologies that people would talk about in this program, I'm really interested to what we call transcranial electrical stimulation. And I know that you know about the technical details about this specific technology, and there are interesting potential for this really scalable and non-expensive brain stimulation technology. But I'm really interested to see how we can kind of use brain imaging methods to see how this technology would work inside the brain and how we can change brain activation and brain connectivity using this technology. But from the first days of doing these studies, one of the things that is making me really not surprised, but make me really kind of interested to explore more is the variation that we see among the patients, among the subjects that we have. As you can see here in this graph, we have kind of sham group and people with kind of the real stimulation. As you can see here in the real group, we have a wide range of variation among the subjects that we have, which is quite interesting in terms of, there is a significant difference between sham and active stimulation, but there is a larger variation among those who are receiving the active stimulation. And there is always a question why people are different, why people are responding differently, and why we have a large variation. And as you can see here, there are people who have more kind of response, and there are people who are responding less to the stimulation. And even when you visualize what we have in the individual level in terms of what are the responses, I'm not going to discuss about the specific outcomes because I'm mainly interested in craving and drug addiction, but I'm not going to discuss about those kind of specific topics. But generally, you can see that there is a variation in response among the subjects. When you do computational head modeling in terms of seeing what is the level of current that people receive when you do transcranial electrical stimulation or TDCS or TACS, whatever technology that you use, you can see that people are different in terms of the level of current they receive. As you can see here, there are people who have really low level of current based on the montage and the brain structure that we have. And there are people who receive higher level of current in those areas. And there's always a question in terms of, okay, how we might be able to consider this individual variation in the brain structure in response to kind of the stimulation that we provide. We have done some kind of initial explorations in terms of is there any relationship between the variation in response that we have and what we have in the computational head model and the level of current that people will receive. It is quite interesting that there are some kind of initial exploratory results that there could be a relationship between those kind of responses that we have in terms of the self-reports that we had for drug craving, specifically in the active group, in relationship to level of the current that the brain receives. and it is not the case for the sham group. So in the sham group, we do not have that level of correlation, which is quite interesting. And even if you see the relationship between structure and what we have in functional responses with fMRI. So when we measure fMRI, there is an interesting response, as you can see here. This is the frontopolar area. Within the frontopolar area, which basically receives the highest level of current in the brain, there is a significant correlation in the active group, and we do not have that significant response in the sham group, which is quite interesting as well in terms of, it seems that there is a response, there is a relationship between whatever we have in terms of the structure and the responses that we get from the stimulation. Those who are interested to see those results, you can find those results in one of the publications that we have done with Maron Bixon's team and kind of Zainab, who was leading this analysis that we have been doing at that period of time. Now we are extending those analyses. I'm not going to go to the details, but it might be interesting that now we have larger databases. We work on the larger database with 60 subjects that we have been exploring. There are some kind of new interesting results that we have with my kind of PhD student, Fadale, that we have been working on a larger database. And we realized that in a kind of a more kind of advanced analytics that we have done for that, we realized that probably voxel-bot analysis is not the best way to do it, and probably network-based analysis would be the best way of seeing the relationship between the level of current that we see inside the brain and the response to the brain. But I'm not going to discuss about these kind of technical details because we do not have enough time for that. But just to have an idea about potentials for considering the brain structure, the importance of the brain structure, and how brain structure would contribute to the level of response that we have to disease here. And considering that the individual variation would be significantly important in terms of how people would be responding to what we have with TDCS or TES. Okay. Now let's go to the next level in terms of, okay, how the brain imaging methods that we use could be helpful to inform whatever we are doing right now in brain stimulation. Let me bring you a few other examples that we have been doing right now. So one of the trials that I'm doing right now is a trial with what we call network level transcranial alternating current stimulation. So what we do, we target executive control network, we target dorsolateral prefrontal cortex, F4, and then posterior parietal cortex, which is on the P4, and we try to do synchronized stimulation or non-synchronized stimulation, see how we might be able to increase activation within executive control network, with the hope that we can help these people to bring back their control and be able to control the drug range. This is an ongoing clinical trial. I was hopeful that I could have the results right now, but I mean, with the pandemic, we have not been able to complete the trial so far. So we are still working on the data collection for that. I'm hopeful that we will have the results by probably early next year. But we are working on this trial. But working on this trial, we have also started to think about, okay, is this the best montage that we are doing in terms of the stimulation? And should we kind of how we might be able to find the best montage for what we are doing? So we have done a new computational modeling analysis in terms of defining, okay, if we put the electrodes on the montage on F3 and F4, these are going to be the major areas that we receive the current side. And with the kind of the parietal electrode montage, these are going to be the areas that would receive the highest level of current inside. And then the frontoparietal connectivity, and we have interest to see how is the kind of the frontoparietal connectivity and how is the connection between these areas. In the first analysis that we have done, we realized that unfortunately we do not have a really good connection between F3 and P3 and F4 and P4. So basically these two areas are not connected as we have been expecting before in terms of kind of especially in the kind of the task context that we are interested to for drug reactivity. So these areas are not connected as we have been expecting before, which was frustrating for the kind of first step. So we have to start thinking about, okay, is there any frontoparietal connectivity among the patients that we have, or should we kind of think about changing the idea of importance of frontoparietal connectivity or executive control connectivity for drug craving? We used the F3, F4 seats that we had, and we have done a whole brain PPI analysis to see if we have frontoparietal connectivity. And we realized that, yes, we do have frontoparietal connectivity during the context of interest for us, which is drug craving. So we do have frontoparietal connectivity. But I mean, those areas in the parietal cortex are not located to what we have been thinking about before. So we checked those areas and we realized that those areas would be mainly under CP5 and CP6 electrodes. And so we realized that we probably need to have an informed montage based on what we had from fMRI in terms of considering frontoparietal connectivity, not on the F3 P3 or F4 P4, but on the CP5 and CP6. As you can see here in the kind of the next levels of analysis, we realize that yes, when we consider the informed montage, we have significant frontoparietal connectivity, and these areas are basically connect as we can expect based on the analysis that we have done before. So I mean, this could be potentially a better area if we are interested to frontoparietal connectivity, and we are interested to see how we can modulate this frontoparietal connectivity using the frontoparietal synchronization methods with alternating current stimulation. The other thing that was interesting for us was seeing how kind of people would have different levels of current in their brain when we do frontoparietal synchronization. With the classic montage, by classic montage I mean F3, P3, F4, P4, if we give 2 milliamp to the frontal area and 2 milliamp to the parietal kind of area, we have almost same distribution of current in frontal and parietal area, which is reasonable. So we are giving the same amount of current in both frontal side and also parietal side. But in the new montage, based on the specific brain structure, the thickness of the skull, whatever we have, with the same level of current in the frontal and parietal side, the parietal side would receive high level of current. So as you can see here, in the parietal side, we have high level of current. And we are talking about 60 patients here. We are talking about 60 subjects that we have been doing modeling for them, okay? So you can see that there is a high level of current here, and there is a significant difference with the level of current that you have in the frontal and parietal side. And if you are interested to have a balanced stimulation in frontal and parietal side, then you probably need to the level of current that you the patient and top of like 2 milliamp in the frontal side and a lower level of like 1.2 milliamp in the parietal side. So we need to change that if you are interested to have balance. So as you can see here, both fMRI and computational head models could inform the way that we define our montages and also the level of current that we use for stimulation, and that is going to be the next level of optimization for whatever we want to do for brain stimulation. That specific kind of pipeline that we suggest based on the results that I showed you, I mean, the pipeline has specifically 8 different steps for informing a montage for dual site stimulation and how we need to optimize even the level of current. I'm not going to go to the details about that, but we have already put that in bioRxiv. So the paper is already under review. So hopefully, I mean, we can get that published soon. But you can find the preprint in bioRxiv. So you can find that in bioRxiv, and you can find the kind of the pipeline that we suggested at that part. Okay. So that was another level in terms of, okay, we can use fMRI to optimize the montage, optimize the the stimulation parameters for what we want to do. Let's go to the next level in terms of, okay, is there any chance that we can monitor whatever is happening with electrical stimulation inside the brain with fMRI? So what we call concurrent fMRI. So we can just see, okay, if we are doing the stimulation, what is really happening right now inside the scanner when we do the stimulation? And we can do fMRI, or we can record the brain while we are doing this. So this is what we call concurrent fMRI. Whatever I showed you till now, there are fMRI before and after stimulation. But now we are talking about just monitoring whatever is happening at the same time of the stimulation. So now we know we can basically control the level of noise that we have inside the scanner when we do concurrent fMRI. So we know that we can control noise, we can control the safety, and there are some technical details to be able to make that happen. So we have been thinking, okay, it's quite important for us to make sure that the quality of what we do is going to be in a level that we can rely on the results that we are going to do. In this context, we realize that there are multiple labs across the board that are interested to combination of TS-fMRI. Most of them are not doing concurrent TS-fMRI, but many of them are interested to basically combining TS and fMRI. So we made a network that we call the International Network of TS-fMRI from these countries, from these labs around the world. And we started to kind of have meetings together and see how we might be able to improve the quality of the field with something like a collaborative initiative. So we made a steering committee of experts in the field, and then we have done a systematic review on the studies that are basically published concurrent TS fMRI results. So we started from this point, and we have done what we called a consensus study with a specific methodology. We brought people together, we worked together to see how we can define a specific quality checklist for what people are supposed to report when they do a TS fMRI study, a concurrent TS fMRI study. If you are interested, you can find the pre-registered report or pre-registered protocol of what we have been doing in OSF. So you can find that in kind of those open science foundation platforms. I'm not going to go to details about what we have done, but we formed a specific expert panel, 75 different people. We asked them to vote different kind of items that we have been thinking that are going to be important. So we developed a checklist of different items that people are supposed to consider in their studies. And based on that, we technically developed what we call contest checklist or concurrent TS-fMRI checklist. So the contest checklist has, as you can see here, some main items to report and also some additional recommendations. So people can basically, we recommend people, if they want to do concurrent TS-fMRI, they are supposed to use these items in their studies. It might be interesting that from these 57 publications that we had at that period of time with concurrent TS-fMRI, and right now we have them like 83, 84 studies. So I mean, the number is growing up really fast. But from the 57 studies that we had at that period of time, when we checked whether these studies reported the items that we have been, recommending people to report, we realized that some of these items are poorly reported in the publications that people have been provided. And also, even if I want to show you in terms of how many of these items are basically reported among these studies, as you can see, there is no single study that reported all these main items that we recommend people to report. And there are just few studies that report something like 70% or 80% of the items. So it seems that we basically, whatever we want to do in the field of kind of transcranial electrical stimulation, we need to increase the quality of reporting and also quality of the research that we do if we want to kind of go to the next step. And the good thing is that kind of this checklist has been accepted just a few days ago in Nature Protocols. The initial draft is available on medRxiv, so you can find it on medRxiv. If you are interested to learn how to combine TES and fMRI together, that is going to be your must to read. So that would definitely be helpful for you to have an idea how that is going to be an interesting kind of reading for the next steps. As you can see here, we already have a large group of colleagues and collaborators that are working with us in that international network. And those who are interested, this is the email of the network. So you can just email us if you are interested to join the network, and we can invite you to the webinars that we have. There is a YouTube channel that we have that you can find the webinars that we have. So if you just go to YouTube and search for International Network of TS-FMRI, you can find those webinars which are basically available. And our YouTube channel is not monetized, so we are not making money out of that. It's just kind of, we really like to have others involved in these projects, and we definitely would be happy to have you as the next generation of people who are interested to these studies. Okay, let me go to the last part of my presentation. Based on what we discussed about till now, the question is, okay, can we get to a level that we basically do personalized protocol for each individual patient? Can we go to a level to be able to use whatever I was discussing about today in terms of computational head modeling, fMRI, concurrent fMRI, and putting them all together and then define personalized protocol for a specific patient and try to use that specific personalized protocol and see how we can improve the quality and make increase the response rate among the patients that we have, and try to work on those who are not responsive. Okay, that is the last part of my talk for today. So this is what we have in terms of, okay, we do a stimulation inside the scanner. This is what we call concurrent TS-FMRI. So this is what we already have. And the question is, okay, if we are doing concurrent TS-FMRI, so we can measure, for example, if you are interested to frontoparietal connectivity, we can measure frontoparietal connectivity inside the scanner, which we call target engagement. We can measure target engagement inside the scanner when we are doing the stimulation. And then we can start to think about, okay, how can I optimize this stimulation? Is there any way that I can basically find specific parameter that would increase the connectivity or maximize the connectivity or optimize the connectivity in a way that would be best for the specific subject that I already have inside the scanner? So that is what we call closed loop TS-FMRI in terms of recording frontoparietal connectivity, stimulating frontoparietal connectivity, and then measuring, okay, how much our stimulation was successful in terms of improving the frontoparietal connectivity as we're expecting. And then based on that, changing the stimulation parameter, whether it's just frequency or the level of current, whatever we are doing. Or even we can change the montage and see how we might be able to optimize the stimulation. And this is what we call closed loop. So we stimulate, we record, we stimulate, we record, we try to find what could be the best stimulation parameter that would give us the highest outcome that we are interested to have. So that seems like a dream, to be honest. So being able to do that inside the scanner. So that is the next level that we are trying to do. To be able to do that, of course, we need to kind of optimize the stimulation protocol. We usually kind of start with stimulating watermelons, and we kind of do the same protocols with watermelons, putting the electrodes. As you can see here, this is the way that we do frontoparietal synchronization. So the multiple electrodes to be able to do high definition stimulation inside the scanner, putting all those things together. As you can imagine, we can just stimulate watermelon, so we can record inside the watermelon, and we can see what is really kind of what do we have inside the watermelon. And of course, after the stimulation, we kind of we eat watermelons. So that is the fun part of the stimulation session that we have inside the scanner. And after watermelon, what would be the best thing to stimulate? Of course, the head of the PI of the project would be the best thing to stimulate. So I would be the first person to go inside the scanner to see the stimulation. and also kind of recording inside the scanner. So we started with myself, and then we kind of extended that to a larger trial that we received a kind of a IRB approval for that. For being able to do that, we have been able to optimize the electrodes that we had inside the scanner. There are some challenges in terms of putting the electrodes in a way that would be working inside the scanner, because you are supposed to put the subject head inside the coil, which is not a really large place to put specifically a large head like my head inside the MRI coil, and how you can put the electrodes in a way that would be functioning, and what should be the structure of those electrodes. So we have developed lots of expertise on that, and we are really happy to share those skills that we developed for doing those things inside the scanner. So with a postdoc and a PhD student that we had at Laureate Institute for Brain Research, so we have been able to make that happen in terms of recording and also stimulating inside the scanner, okay? Which is not very much easy to do, but technically it is feasible. And this is just to give you an idea about how it works and how is the structure of what we do for closed loop stimulation inside the scanner. So this is the protocol that we already have for a trial that we have recently finished. Now we are doing data analysis for that. I will tell you some good things about that. So as you can see here, we do some optimization phases. We stimulate and record and stimulate and record to see if we can find the optimized stimulation. And then we use that optimized stimulation to see if there is a significant effect between optimized and non-optimized stimulation. So this is the kind of design that we have inside the scan. And if you are interested to know a little bit more about how we do a stimulation and recording, so what we do, basically we have blocks of 30 seconds, okay? And in each block, we do a stimulation for 20 seconds. So we stimulate for 20 seconds. And then we start to record fMRI response to that with 6 seconds of delay. As you know, there is always a delay between the actual response and the bold response to what we have. So we record frontoparietal connectivity with a 6-second delay. And then for the next round of the stimulation, we have something like 4 seconds here, as you can see here. So we have 4 seconds. What we do, we measure frontoparietal connectivity, we collect the fMRI response, measure frontoparietal connectivity in just less than a second. And then for the do the rest of something like another 1 second, we do the optimization, and we inform the stimulation device in terms of what will be the frequency and the phase of the stimulation for the next round. And then we do the next round. And so we are able to do the online recording and online stimulation, which is what we are interested to. There are some other technical details in terms of testing. I'm not going to go to the details about that. There are always questions in terms of what should be the control condition. There are many variations that we can consider for defining the control condition or the sham condition. I'm not going to go to details, but there are lots of interesting things about that. Those who are interested to know about those technical details, we put that something like a technical report about what we are discussing about in medRxiv. So if you search that in medRxiv, you can find that in medRxiv. We provided all the technical details I discussed about today on that medRxiv pre-registration paper or preprint paper. So you can find that paper there, and that could be a good starting point if you are interested in this technology. Hopefully it will get published soon, and we can have the actual peer-reviewed version as well. We have some pilot data. I'm not going to discuss about the pilot data. We are working on the next publication. I hope that it will be something interesting to explore. Just to wrap up what we have been discussing about closed loop stimulation, let's give you an idea about the potentials in the field. Let me just give you an overview about what we discussed about, and then conclude and wrap up the session. So as you can see here, that we do frontoparietal connectivity, we need to measure what is happening inside the scanner, and we have a sliding window in terms of measuring the frontoparietal connectivity, and we measure the target engagement. One of the major challenges that we have is to define the temporal dynamics of this response. How long should be the sliding window? How long should be the block? How long should we stimulate to get a reasonable response from the brain that could inform the next round of stimulation? So that this is one of the major challenges that we are already facing. And then what would be the best optimization algorithm that could be used? So there are still many challenges for that, and we are working on that. Okay, so let's wrap up. What we discussed about today, we discussed that people respond differently to TS. So that is one of the most important things that you should consider when you do a trial with TS. There are important functional and structural items or factors that could contribute to the inter-individual variation in response to TS. We discussed that fMRI could be used to inform optimized montages and optimized montage selection. We discussed that the level of dosing of electricity that would put inside the brain, or over the brain, or over the skull, could be optimized with using computational head model. As we have discussed, the concurrent TS fMRI could be informative. There are interesting things that could be done with concurrent TS fMRI. What I can tell you right now is at least closed-loop TS fMRI is technically feasible. So this is something that we can do, and it's technically doable, and there are other labs that are trying to do that. So it's going to be feasible. And there are interesting potentials for international collaboration. And I'm always kind of impressed by what Felipe and his colleagues are doing in terms of bringing people together and help them to collaborate. So I'm really open for further discussion in terms of how we might be able to help other labs around the world to do the same thing. Those who are interested to these areas and cognitive neuroscience, I have already put a new course on cognitive neuroscience online on YouTube, which is publicly available. It has like 71 videos, so you can find those videos online. And my channel is also not monetized, so I'm not making money out of that. I really love that, and those who are interested to know about those technical details, you can find kind of those educational courses there are available. So I need to thank my colleagues, collaborators, brilliant PhD students and postdocs that contribute to these studies. I'm hopeful that I will be able to kind of help others to do the same in the future. Thank you very much.
So Hamed, can you share the link here for your? Oh, you did. I think Sarah did share the link for your neuroscience course. That's wonderful. I did not know that. We'll post that as well for other colleagues. You do have I'm thinking as well. Some of the audience here may want it. Do you have captions there in the videos? No.
No, we can basically do that if there is anybody who is interested to help us to make captions. Yeah.
And that's one thing that Hamed also, we've been working together, and that's the goal. That's how we can do things in science, achieve big projects. And so if you all interested in learning more, and so that's a wonderful person to touch base and do projects in collaboration. So anyway, but I'll pass to Pablo. Pablo, sorry, I keep cutting you off. Pablo,
no problem at all. So Dr. Hamed, we have a few questions here. One is from Alejandra, and she says, "Hi, Dr. Hamed. It's a very interesting lecture, and yes, indeed, so far, the noise is one of the main problems, but sounds promising. One question is regarding the combination with TS, as it is brain state dependent. Then how does your group manage anxiety or an attentional state of participants inside the fMRI when combined with TS?"
Yeah, that's a brilliant question, and I loved it. So there are definitely lots of challenges. One of the things that would be interesting for others to know that it seems that the response to TS or any sort of brain stimulation that we have could be state dependent. And what do we mean by state dependent? By state dependent, it means that if your subject would have a specific functional state right now, for example, your subject is anxious, your subject is exhausted, your subject is fatigued or depressed, whatever, or what would be the functional state of the brain right now would contribute to the level of response that people would show in in response to electrical stimulation. And it's important to control that specific condition. And meanwhile, what we can do, we can also take advantage of what we do with the brain stimulation, with closed loop brain stimulation. The good thing about closed loop brain stimulation is that we basically take all these factors into consideration because we are monitoring what is really happening at that period of time. We are, whatever the state is, we are considering that state, and then we record the response, and then we optimize the stimulation accordingly. So that is the advantage of what we have with online stimulation recording system that I'm talking about right now. So there are specific advantages for that. But the protocol that they have, they expose people to smoking-related cues, and they get people to a level of activation in their brain that make the brain brain state ready to be responsive to the stimulation. So that is going to be a sort of a good factor in terms of putting people in that specific state that would make people more responsive to the stimulation. So brain state is an important factor. It could be a confounding factor in the studies that you do. It could be controlled with closed loop brain stimulation, but meanwhile, we need to take into account that the current brain state could also be could be beneficial for you if you want to do brain stimulation in the clinical setting. So that might be even helpful for what you are interested to do.
Thank you. Audrey Sartori has another question. He asks, can the electromagnetic field of the resonance interfere with transcranial stimulation, affecting the amount of current that reaches the cortex?
That is always a challenge and a challenging question in terms of, do we have how is the relationship between whatever we have with fMRI and with electrical stimulation? So we know that there is an interference. So we know that there is an interference. But what we know so far, first of all, what we do most of the time, we use the fMRI pulse sequences that are technically low energy. So if we use really high energy fMRI sequences, that might make things a little bit more complex because then the interference would be higher. So we use low energy fMRI sequences. And then based on what we already have, and we have done different studies, and even different labs, they have done the same thing. It seems that the level of interference would be not beyond few millimeters under the electrode over the skull. So we are very hopeful that the interference would not be significant beyond that point. So whatever we have over the cortex hopefully would not be affected, at least based on the empirical data that we already have. So we are pretty confident that we can do concurrent TS fMRI inside the scanner. And as you know, it is not the case with EEG. With EEG, I mean, most of the labs, their conclusion is, so far it is not technically doable to do concurrent TACS EEG because of the some challenging technical issues. But for fMRI, it seems that we can technically do that inside the scanner.
Perfect. And we have just another question here from, Dr. Danielson Juan. And he's asking, how are included structural brain lesions in the electro-brain model for TDCS?
Yeah, that is definitely something that people have done. I have never done that in my own lab, but people who are doing these sort of studies, especially with the stroke patients, they have done, lots of computational modeling showing that how it is going to be important to consider brain lesions. What we do, I'm mainly focused to substance users. For substance users, we know that they can have brain atrophy, specifically in the frontal areas. So, the routine computational models take that into account. So we consider that, and that is one of the factors that we consider in the solution. But beyond that, we have a larger stroke-related lesion in the brain, then there are specific modeling issues related to that, which I'm not really expert on that, but there are many publications using kind of computational modeling to consider that in the stroke patient.
One last question. I think we have time. So Sara Barbosa is asking, Dr. Hamed, in your studies, you usually use stratification by substance use, or it is not real important at a time?
It depends on the level of resources that you have. I mean, it's always a question about the amount of money that you have. What would be the size of the study that you want to do? I've not been very much kind of entrapped by clinical variation among the subjects, but we always consider that as something like an important potential predictor in response. As you can imagine, when we have substance users, for example, I'm mainly going to work with people with meth use disorder or stimulants. Sometimes we have people who are using a stimulant with opioids, and sometimes we have people who are using a stimulant alone. So there's a question in terms of, okay, do we have different responses among stimulant users who are using opioids or those who are not using opioids? So that's a relevant clinical question. And it's always an important issue in terms of whether it's something like an exploratory analysis or it's a confirmatory analysis, because there are many factors that could be contributing to that. And I mean, how much exploration we can have in a database with 30, 40, 60 subjects, just we know that they are going to be limited. One of the interesting things I'm realizing that is going to be definitely important, people reported that in different studies, at the first level is basically gender. Gender, it seems that it's a really, really, really strong factor in terms of how TES would be important or would be kind of changing the brain. So, and even when we talk about kind of things like gender, there's always a question in terms of, okay, if you include women, how you can kind of consider their status in their menstrual cycle, how can you consider level of estrogen, progesterone. So it's always a challenging question in terms of what are the factors that could be controlled in the further analysis. But I mean, the good thing is that you technically record whatever you can record, and at the end of the day, you put them in some predicting models to see if there's something significant would come out of that. But there's always a challenge in terms of how much power and degree of freedom that you have for these exploratory analysis.
Thank you again, Dr. Hamed, a wonderful lecture.
Thank you very much for inviting me, and hopefully it would be a really fruitful kind of meeting. And I'm really hopeful that we would be able to see each other in person again. So I'm looking forward for that in 2022, hopefully. Yeah.
We hope the same.
Thank you so much, Hamed. Likewise. Hope to see you soon. Thank you.
Thank you. Bye-bye.